So now we have a special presentation about research updates. I think this is one in which many people are curious, and we have an outstanding woman, Dr. Amber Van Laar, and she is a movement disorder specialist neurologist and a passionate researcher. She currently serves as the Senior Vice President of Global Clinical Development for AskBio, a biopharmaceutical company. I think some of you know her, and let's welcome her. She's going to talk about gene therapy in particular. Thank you so much.
Thank you. See if I can see. So yeah, I think I know many people in this room in a variety of capacities, but today I'm here to talk about an update on our gene therapy. Can you not hear me? Louder? Better? Any better? Okay, there we go. So I have a lot of love for research, as you just heard. I can talk research all day long. If you want to come find me, if there's something else you're kind of curious about, I have something probably to say about it. But today I'm going to talk about gene therapy. As full disclosure, I'm coming today as an employee of AskBio. It's a gene therapy company. I'll tell you a little bit more about at the end here. But just to give a gist of what I want to talk about today, there's actually a lot of data in here, so we're going to kind of walk through that a little bit slowly. But first, I want to say, why am I even up here talking about something like gene therapy for Parkinson's? There's still this massive unmet need. I want to talk about what gene therapy is, but I also want to say what it isn't, because I think there's a lot of misunderstanding out in the world. I wanted to talk about what it means to do gene therapy in the brain, because this is kind of unique and can be a little bit scary. But why do we want to do gene therapy for Parkinson's in the first place? And I want to give you an update then on some data that we have right now that's about to be published here any day now from a study that's just wrapping up. And then we have a new study. That's actually the other big thing I wanted to bring up today. So we have a phase 2 study that just opened up, and we'll talk about that. Okay. It should be no surprise to anybody in this room that Parkinson's is very common. This is increasing. All the data that we're seeing right now suggests that Parkinson's is just getting bigger. Lots of reasons for it. We just heard about the registry. Maybe some environmental exposures and things like this may be part of the reason why. Whatever it is, Parkinson's is getting bigger and bigger and bigger. What are we going to do about it? And this is part of what I want to talk to you about today. I think I just want to point out real quick, so men and women both obviously can get Parkinson's, but there is still, as we've seen before, a little bit more number of men get PD than women. Parkinson's moves on. It just keeps going. And for a lot of people, after even a couple of years of living with the disease, they're starting to already have problems with the medicines that we have at hand right now. And this means that they start to feel the medicines they can wear off. They're not maintaining the good day. The predictability, like you take the medicine, you know it's going to work, how long it's going to work, that starts to dissipate over time. And in short, really, we have all these medicines, and yet there are still a lot of shortcomings to what we have available today. And in spite of all of that, the disease still progresses. It still goes on. We're doing nothing to actually slow down disease progression to this day, in spite of knowing about Parkinson's for a millennia. So we still have fluctuations in medications and how this gets, how a person responds on a day-to-day. And just the ability to have one full good day, that starts to get harder and harder as time moves on. There's a lot of other non-motor symptoms. I'm sure there's going to be lots of talks about that today. There's a lot of things that people can't see, and often this shows up first and can be a bigger problem for people living with PD than the stuff that people can see on the outside. There's not much out there to address it, even now. So deep brain stimulation was incredible. Like, this was a big game changer. For me, practicing, this was the one thing I could tell patients, I can really make a difference in someone's lives with something like DBS. But even with DBS, there are still some limitations, and it doesn't actually treat the underlying causes of Parkinson's. And so at the end of the day, in spite of all this work and all the research that's been done, we still, to this day, the best advice that we can give is exercise to slow down disease today. So I got really tired of just changing levodopa dose and continuing to tell my patients to exercise, so I left the clinic and continued to do some more research. But I think it helps to understand, so gene therapy has actually been around. This is, I think we're going into our second decade of gene therapy for Parkinson's. Like, what happened? Where did this all go? And so I think the big part of there's some big questions that got left behind. So did we go to the right part of the brain? Did we get the right type of Parkinson's in that patient population? Did we get the right stage, the right patient? And then at the end of the day, like, how do I not only show us, but the patients, their families, the FDA, and other insurance companies that what we're doing is actually starting to alter the course of disease? Major, major, major challenges that we're working with. Now, the reason I got excited about it and I made a big change to go into this, because I really think gene therapy has potential to do things very differently to really get at the core, the underlying causes of Parkinson's disease. I have to put my sunglasses on here, I can't see. So right now, as I said already, Parkinson's, all the medicines that we have out there now do nothing to actually treat the underlying causes of disease. One hope with gene therapy is that this is durable. It's a one and done. We do this once, and then there's nothing else to do other than just manage how the disease continues to change or not over time. And really, at the end of the day, the reason I got excited about this is that we hope, and what I'd like to show you today is that we think that GDNF, which is the gene therapy we're doing, may actually get at the root causes of the disease so we can actually stop it in its tracks. Okay. Now I have to take you all back to school here for a little bit. So some of you have seen this before. It won't hurt to hear it again. Well, we're going to do a little bit of gene therapy 101, but I have to just start at the basics here. So what is a gene? A gene is DNA. DNA is our genes, and it makes up everything that makes us look and behave even the way that we do. We know that there are lots of genes that we share. Our genes we are mostly the same, and we share a lot of genes even with the Tyrannosaurus rex. It's about 75%. And it's really only this 1% of genes that really makes up the difference between the way that we all kind of look. It's just really that 1%. But what just kind of go back to basic biology here. I'll have to use this screen. But we have a cell, and inside the cell we've got DNA, and you see the X's, the chromosomes. We have a whole bunch of those, and within those DNA, there's little segments of the DNA. That's our gene. And and from those genes, the machinery inside the cell reads that and makes a protein. And it's really the protein that does the work for you. And this is a common thing that happens every day in all your cells all day long. When we're doing gene therapy, we're basically taking advantage of this happens every day in all your cells right now. It's happening. So we're taking advantage of that cell being able to read a gene and turn it into a protein. In the case of gene therapy, we're using our genes like instructions. So we're saying, I want this gene to be made a little bit more in this part of the brain. And what we do is we put a little piece of this DNA in so that the cells can pick it up and read it and then make more of the protein that we want. In our case, it's GDNF. I'm going to keep using that word a lot. So GDNF is our really the medicine that we're trying to use. And so we're using gene therapy to take advantage of what's already happening naturally to then ask the cell, please make more of this protein. So what does this kind of look like? So this is really what the gene therapy looks like in a really kind of cartoonish way. But we have an outside, so we have a capsid, this is our vector, it's our packaging. And inside then we have a little piece of DNA, our genes. It's our gene that we want the cells to make more of. And it put all of this together, and that is what makes a gene therapy. So there's more. Break this down a little bit more. Okay. So it helps to think about our gene. Think of it like a letter. It's a message you want to send to somebody. So we got our DNA and our letter. And just like when you want to send somebody a message, you got to put that message into an envelope. And that's what we do with gene therapy. We put the gene into our envelope. In this case, it's an AAV. And then we've got to get that delivered to the right part of the body. In the case for Parkinson's, we're doing delivery right to the brain. And so we have to get our envelope and our now sealed, signed, put the envelope on, put the stamp on, and now we got to get delivered. And in our case, we do it in the brain. And then once it's in the brain, the cells do this all the time. They're really good at taking up that vector, so they pull in our gene therapy, and it takes that envelope just like you would do it at home. You take an envelope, crumple it up, throw it away, and all you've got left behind now is the message. And in our case, all we're left with is just this little piece of DNA, which is for GDNF. And then we ask the cell to do what it does every day, read that little message and make some of the instructions are to make more GDNF. Okay, now here's a cartoon version of that. Here's our DNA. This is our gene, and in our case, it's GDNF. And basically in a test tube, we take that, put it together into our package, our envelope for the AAV. These things come together just on their own. This is sort of a natural process that happens in a tube. Now, once we get that all made, we put a whole bunch of these little vectors into a part of the brain called the putamen. There's videos in a minute for you. And the cells take up our vector, and again, they take that envelope, throw it away, and all you're left with now is that little piece of gene, and we say, cell, please read our gene and make GDNF. And in this case, this is what's happening. So the green dots here is meant to represent a lot of GDNF being made and then shared across. And this is actually a big benefit of this gene therapy because it's not just the cells that pick up our gene, it's actually all the cells around it that also benefit because GDNF is then secreted and we get the benefit in a lot of places this way. So it's pretty clever. So why would we think about gene therapy for Parkinson's? A lot of people would say, where would you start? Like, why would you even think about this? And I kind of laid it out early on. I mean, we have a lot of we have medicines, been around since the 60s, maybe in some cases. We're still using them. There's still a lot of problems with them, and they're still not trying to actually get at the bottom of the disease. I also bring this up because gene therapy is not just for purely genetic diseases. I think we often see in the news if it's gene therapy, it's probably in a rare pediatric condition. But I'm trying to upend that and say that we can actually use gene therapy to treat even large diseases, something like Parkinson's disease. And because to think about doing this so that we can get at the bottom of it and really start to treat some of the underlying causes of Parkinson's. And the other big benefit to something like gene therapy is one time. So here's kind of how GDNF has evolved. So I've had the good fortune of being exposed to GDNF for many years, working in the lab. And I saw it even back in the 90s, like, man, we may have found it. We may have found it. And even back in the 90s, the question immediately came up as well, how do we get it in the brain? And here we are, 30 years later? And I think we finally figured it out, but it took 30 years for us to do this. And so you can this timeline here is even really abbreviated to just give you a sense of it. It's been decades of what data I'm about to show you took decades to get us here. So GDNF and PD, how do these things go together? Maybe some of you have heard of GDNF before, but we learned back in the 90s that GDNF is required. You must have GDNF for those dopamine neurons just to develop. You don't have GDNF, you never develop dopamine neurons, and you never survive. So we also found that people with Parkinson's have less GDNF. So this idea that perhaps lower levels of GDNF it's one of the things that sets up the whole environment to make it more likely that those neurons are going to die. And so the idea came then, well, if we gave back more of that GDNF is a growth factor, a neurotrophic factor, meaning it's food for neurons. If we give that back, can we make those cells that are sick, can we make them healthier again? And can we stop the other cells from dying? This is what we've been trying to pursue. It's been 30 years of trying to understand how GDNF works for Parkinson's. So again, can we use GDNF as a way to really help the sick cells, but also prevent other cells from dying? And that then has the potential to actually change the course of disease. And that's what we're trying to ask right now in our clinical trials. Okay. So my mom, how do you do this? How do you do this? Okay. I'm like, okay, Mom, let's just sat down. And this is pretty much how I described it to my mom to try and help her see this. You need to see it. So we actually are doing this in an MRI machine. The entire procedure is done in MRI, and that allows us to watch very closely, spot on. We are on the right spot, we're on the right target, everything's okay, and we can see in real time if anything else happens. And so we have the MRI machine running the full time. Simply getting us to the point where we could perform a safe surgical procedure in an MRI machine, that alone took over a decade. But we got us here, and that's how we knew that we were definitely getting in the doing the delivery part right. Because otherwise, it's a big black box. Whenever we put leads in before, we were doing anything in the brain, we really couldn't see. This is the first time we're able to visualize in near real time what we're doing in the brain. So that's a big leap in just that technology. So right now, our studies are a lot of screening. We want to make sure we've got the right patient for this procedure. We do a lot to make sure people are safe and would qualify. After that, if people qualify, it's a one-time procedure. So yes, it's brain surgery. This is similar in a lot of ways to something like DBS, but it's one and done. There's no hardware left behind. It's a one-day procedure. Most of our participants were able to go home the next day. This has been a technique that's been used in a variety of other conditions. So for epilepsy, for brain tumors, the same sort of technique. This delivery is very similar. And it's been done for over 15 years in the brain. So there's some history here, but applying it now to something like Parkinson's is not newer, but a little bit newer. So this is at least a cartoon version of what this starts to look like. So essentially what we're looking at is placing a hollow tube in the back of the head and slowly infusing and advancing this into a part of the brain called the putamen. So we want our gene therapy to go in that part that's green, and only that part. That part's called the putamen. So that's right, pretty deep inside the brain. So it's not too different from places that we like to target for something like DBS. It's not too different. This allows us to give an infusion that is customized to everybody, not even everybody, every putamen. So each side of the brain, we customize those infusions for that. And this is all done then under anesthesia. No one's awake for this one. So this is what this looks like so this is now sped up. This is one of our procedures from the study I'm about to show you here. So let me try and point this out. So eyeballs up here. Ears would be the sides here. This is the back of the head. This black line you can see there, so that is one of the hollow tubes that's going in the back of the head. I'll do it over here. So we've got the black line here. So that's one of those hollow tubes I had mentioned. So here's eyeballs. The ears would be on the sides. This is the back of the head. And what we're seeing here is the white that you see coming in. That is actually basically a dye that's been added to the gene therapy, and it allows us to see it in an MRI, so we can see exactly where it's going in near real time. Now this is sped up several times, so this actually takes several hours to do. We've sped it up just for a video for a demonstration. But what you're seeing happening here is the surgeons are slowly inserting. There's another one of these hollow tubes on this side. We just can't see it in this particular cut. But they're slowly infusing this white, which is the gene therapy, and they infuse and they try to fill up just that part of the putamen, and then once they fill that up, they move forward, and they fill that up, and they move forward, and then fill that up. And this allows us to try to fill up this really kind of awkward shape. It's like a comma. So trying to fill up just the putamen and only putamen, this is the technique that has been developed, and again, like 30 years to get us here. But we learned that it really does you have to get a good spread in the brain region, because I need more of those cells to pick up that gene to then make more of that GDNF to really get the benefit from this. So, complicated, highly technical. We're doing a lot to try to make this a lot faster and smoother, but yeah, it took us this long to get here, and I would like to argue that it's working, and I'll show you why. So, again, one day. It's surgery. One day. One day. You don't have to come back for getting the batteries changed. There's no manipulating with the voltages and changing. It's just one day. Everything else after that is managing your meds just the way you would normally with your physician. There's a lot of visits. You can see how technically challenging this is, and we've got a lot that we have to show to do things. People like the FDA, so it's very rigorous. These are difficult studies, but. I'd like to think, and people who join these studies do it because they want to give back to the community in a big way. So a lot of visits, especially in the first 3 years, it does ease up over that, where it becomes more annual type of visits over time. But the goal is that we want to follow people for a really, really long time, and the FDA is also asking us to do that too, because it's gene therapy. I think that's fair. So that's sort of the gist of how that part happens. So I would really like to show you some of the data that we're getting from our phase 1B. This is, I hope, going to be published in any day now. So there's a little bit of data. So this is looking at our data from up to 18 months. We now have up to 4-year data, 3-year data we've now presented. But this is what's about to be publicly published here. So we had 11 participants that were in this study. This is a phase 1B. Just a couple things to highlight here. We split the groups up into two. We were interested in looking at someone with really early stages of Parkinson's and someone with more middle stages of Parkinson's. And did we see differences in how they reacted to our GDNF? And so there were some differences in the groups, but in places that I wanted it to be, in places and not different where I didn't want it to be. I want the ages to be the same, good. I wanted the men and women ratios to be the same, good. The time from diagnosis, though, that's different. So on average, our mild group was only 2 years from diagnosis. versus our more advanced group was about 7.5 years from diagnosis, right? So just kind of give you a sense. I know many of you sitting in the room, like where would you kind of fit in that category? And that means their UPDRS scores were different as well, and how much levodopa they were taking was also different, which is good. That's what we wanted. Okay. So we did our 1 day of surgery. That's the video that we saw earlier. So you can see these bars shouldn't look very different, and that's great. I want them to be the same because that means that for the very first time of doing gene therapy, putting drugs into the brain like this is the first time that we've been very consistent, and there's not been the delivery been a question of a variable in the outcomes. So this was good. We got a lot delivered. This was the maximum that it's ever been reported to date. So this has been really exciting for us to finally get this one variable down. So this is a look at safety. So AEs are adverse events. This is what did we see? So there's going to be adverse events in anything that you look at, even aspirin. But what we saw is that there are things that happen right around the time of surgery, and then there's pretty much everything else. So around the time of surgery, we had a lot of patients that their shoulders, hips, legs from laying on the MRI bed for a long time. That was one of the most common things that we saw. Headache. The holes are drilled into the brain. We had some headaches, things like this. But nothing that was unexpected. All these things were they didn't last very long either. It was Tylenol and they went home and it went away. Some things that we did have that did show up later. So we had some COVID infections as we were doing this in the time of COVID. But really we had someone with a little bit of dyskinesia that popped up. We also consider that a great thing. But overall, this is really benign for a surgical type of safety profile. So what did we see? So this is what we're really excited about. Okay, I'm gonna have to walk everyone through this real quick. So this is some good data. So red is UPDRS part 3. So this is when they're measuring how fast and how big you can move. We get a score from that. And that the bigger the score, the more severe and more the worse the Parkinson's symptoms are. Okay. So the red line is I'm measuring that UPDRS without medicine. So how bad is it with no medicine on board, no levodopa? The green line is I'm now going to check that again, but with levodopa. How much better do you get if I give you levodopa back? The blue line is looking at UPDRS part 2. So how hard is it to cut your meat using utensils, brushing your hair, brushing your teeth, shaving? That blue line represents those kinds of things. And so let me walk you through what we saw in the mild group. So our mild group is here on, I guess that's your left. And what we saw is a lot of no change. I actually think this is great. I would love to see this line stay flat for 5 years. That means we have somebody that was diagnosed 9 months ago, that their Parkinson's is no different today than it was 5 years ago. I think that's incredible. I need to wait to see that, but this is where it becomes a challenge to show like no change is amazing. that is one thing we're really going to be facing here. However, we saw that in our moderate group, so this was a group of patients about 7.5 years from diagnosis. They're on a good bit more of levodopa. What we saw is their off scores dropped by almost half. It took about 6 to 12 months, so it takes time for us to see this drop. So this is our moderate group here. So again, this is that red line's their off score. So we saw from where they started to about 18 months, which was our primary endpoint. This is when we wanted to look. It was almost half of what it was when they came in. What I think is also really cool, though, is that their off score is now matching what they're on, as good as they could get on with levodopa. That's what their off score looks like now. And the way that it looks to me is if we're kind of turning the clock back on their Parkinson's and getting to a place where it's easier to manage, more predictability, longer lasting. And so this is exciting. So, okay, it's one thing for us to have, the UPDRS is rated by a neurologist to get a number. That's one way to look at this. Another way that we need to look at it, though, is with diaries. These are really awful. They're annoying. Every 30 minutes, are you on? Are you off? Are you having dyskinesia? Are you asleep? We ask our participants to do this with a lot of regularity. This is also very important to the FDA. And we said when you started, we're looking at the green here, so that's your good on time. That means you've taken levodopa, it's kicked in, you're feeling good. You may have a little bit of dyskinesia, but it's not bothering you. So we want more green, less yellow, less red. And sure enough, that is what we saw here. So when they came in, they had a lot of green, but a lot more red and a lot of yellow. And over time, by the 18 months, we see almost none of the yellow, meaning that yellow being time with dyskinesia that really bothers you, that basically dissolves. And the red is off time. So that's when you may have even taken your meds, but you're still off. It's still not really kicking in. We were able to see that go down as well. So we're seeing better off time, less time with bad dyskinesias, and more good on time. This is encouraging. So patients are giving us information back that what we can see, what we can rate with that number, matches what they're also seeing and rating themselves at home. So that to me was really remarkable. So I could go on and on about this forever, but I mean this is exciting. We wanted to know what GDNF would do when delivered finally in the right amount, in the right way, with enough time and in the right groups. And this was that first study to really help us understand that. We need more time. This is uncontrolled. I mean, we don't have a sham. There's no control for us to compare this to. That's a major gap. But that is the study I want to show you next. And at that mild group, I need to see what they look like out at 5 years, because that tells me something very different than seeing somebody improve in the moderate group. And so, at the end of the day, we needed a blinded study, and that's what led to this. So this is just opened up. This is our phase 2. This is the first time now that we are asking GDNF in a very blinded, truly strong scientific integrity, what is GDNF doing? Is it actually going to alter the course of the disease? This is it's a big undertaking. So this is really the basics of the design. So we have patients come in. There's a lot of screening. We do a lot of, it's a lot of imaging, a lot of testing. There's a lot of blood work. I mean, again, we're trying to really change the course of the way that we manage PD. So this is why we do it. This is a blinded study, so we split participants into either the active treatment or a control surgery group. I can go into this at length if anyone's interested. But this is a double-blinded follow-up. This is the best way for us to go back to the FDA and the EMA and others and say, this is really what the drug is doing. This is what our gene therapy is doing. And once we finally get everybody through everyone's treated, we can then unblind, and then we can offer anyone who was in our control group will then be offered active treatment. We're also randomizing the terms where we split them into the groups. We're doing a randomization of 2 to 1. Meaning that, so that's what this little circle is here. So meaning that for every one person that gets placebo, 2 will be in our active arm and receive active drug. We will then follow people for a long time. We need to know what happens long term after we've done this gene therapy. Right now, we're mandated to do 5 years by the FDA. Our plan is to do much longer than that. We need to see what happens to our patients. This is just rough breakdown of key criteria. I'm here. I've got a few friends from my company that are here as well, standing in the back. So if you've got questions, there's more here. This is a brief overview. But 45 to 75 years old is what we're looking at. You can see the time from diagnosis. Again, we're looking for sort of that middle road of Parkinson's disease. Need to be on levodopa, because this is something we're measuring and watching. What we're not looking for is it's not Parkinson's. That should be obvious. If there's a clearly genetic cause for your Parkinson's, this is probably not the right study for you. If there's a lot of memory issues or psychiatric issues, this is just not the right study, and that's okay. But there's a bunch of, and if there was DBS, if you already have DBS, this would also be a disqualifier. So, again, this is only a quick overview. If you have any interest or any questions, please come find me or one of my colleagues, because there's more. But many people are interested to know where. So this is where we are in the United States. So we tried our best to cover. But what this represents also is a lot of big Parkinson centers across the country. It's like UCSF, where a lot of this was born out of, for example, and we're at MGH and Harvard and things like this, because this is where the experts lie. And this is where we've been training our sites. So if you're interested at all, number one, talk to your neurologist. I hope everybody here has a neurologist. Please talk to your neurologist or your PCP, anybody. Just let them know you're even interested. The Fox Trial Finder, I do like this quite a bit. This is a little bit more patient-friendly type of look. It's better at, if you're in a region, they can help you find trials that are in your area or things that you're maybe interested in. So the Fox Trial Finder does a really good job with that. But we also have friends here. We have brochures, so come visit we have a table out here. But we also have this QR code. So this, if you are savvy enough to pick this one up. So this QR code will take you to a link to one of like a launching page for that phase 2 study if you're interested at all. So you can at least take a look and see if you want to learn any more information. You can do it more passively that way. We're on clinicaltrials.gov. If you are worth your weight in salt, any clinical study must be on clinicaltrials.gov. So if you're questioning if a study is legitimate or not, go there first, and then also check out the Flux Trial Finder. If you have any questions about, you don't want to use QR code, you can ask first at AskBio.com. That also works. And many people here know how to find me. So just real quick, so who is AskBio? So AskBio has actually been around for over 20 years. Of the gene therapies that are approved, that technology was based on work that AskBio had done, which I think is pretty cool. So we're actually quite large, over 500 employees. We cover both Europe and here in the US. Our closest office is in Columbus, Ohio, but our headquarters is down in RTP in North Carolina. A lot of technology, a lot of that we know and working with today in gene therapy really came and stemmed from Jude Samolsky, who was the founder of our company. We are very dedicated to making sure we include patients in everything that we do. And some of you here have probably participated in some of our ad boards and other things that we do. We can't move forward without getting support from the community as well. So please, this is critical for us, and we'll be asking for more. And as I've already said, we're a leader in gene therapy, and we have not really been that way for Parkinson's. I'm trying to really change that, and just part of why I'm here talking to you today. But please, Kara Wyckoff is standing in the back. She's our patient advocacy, and hopefully some of you have seen her in the years past, because we try to get here every year. At the end of the day, thank you for listening. I hope there's more questions. You're free to use this QR code. But I can't be here and wouldn't be up here today if it weren't for the brave people who were taking part in this crazy gene therapy experiment we just did. So big thanks to all of our participants now and earlier, and those that may be coming. So thank you very much. Questions? Now we're doing. Yeah. Okay.
Yes.
Okay. Oh, yep. Oh, I could go on forever and ever about this. So it is not directly impacting my work now. However, all the work that we're doing now came from NIH funding. So we wouldn't be here today if it didn't come from there. So I will probably keep it at that, but it is a scary time. Yeah. Yeah, sure. Right now, it is after a longer period of time. This is as much as I don't like that. There are real reasons for this from an FDA perspective, because it's a safety thing. Having blinded safety data like this is critical. It's Parkinson's, and they look at it a little bit differently because there's already a treatment out there for Parkinson's. So that having that blinded control data is critical. If there's any way to cut that back, I will try to do that. So it is fairly set, but I wish it were cleaner. So I have to get everybody treated, and they have to get through their last visit, and then we can unblind. And so that depends on how fast we can recruit. Yeah. No, please. Thank you. So the question is, like, for the control surgery, what does that look like? So I realized we didn't have a slide on that. So it is basically anesthesia. So you are unconscious because you can't know what's going on. We have people laying in the same position because that also can be unblinding, because you should have like some shoulder pain and knee pain and things like this. So we have people staying in the MRI machine at that time, and basically they sit around the room and watch movies. So there's there is a partial borehole that's drilled, so it's enough to cause like the sensation that something was there, but we don't actually go all the way through. Nothing's inserted, nothing's injected, nothing. So dura, so that is that big tough covering of the brain. Nothing even crosses that. So it's really the anesthesia bit, and we have to do that because it's the best way to mimic what's happening for the active patients.
We have a question over here.
So, yep.
Are there any other gene therapies out there for Parkinson's, and how does that compare to your product?
Thank you. So I appreciate you asking that. So I've been involved in several of them at this point. So we were the only game in town until just very recently. So there was another company that announced that they're trying to start a phase 2. But there's a gene therapy for GBA specific. So if you had a GBA mutation as the cause of your Parkinson's, I believe that study is still open with Eli Lilly. But there's really not much out right now. This is really it.
Is there any specifics for screening? What are the specifics for screening?
Sorry, okay. Yeah.
What are the specifics for screening?
So yeah, the screening is there. It's a lot of the visits are kind of the same, but we need to make sure that you meet that UPDRS amount and that you improve after a levodopa challenge. So just like with DBS, you come in off. They do the scores, you take the medicine, and they do the test again. We're doing the same thing here. I need to see that you are responding to levodopa, because if you have a good response to levodopa, you're probably going to respond well to the GDNF. So it's a lot of imaging. So there's an MRI. There's also something called a fluorodopa scan. So this is a way of looking at the dopamine system in the brain. And then we have another one called FDG. So it's a lot of imaging. But again, this is all set up in order for us to have evidence to prove to the FDA that we're changing the course of disease. So it's a lot of clinical assessments. There's some written stuff, like on a tablet, and then blood work and imaging. I think we have a good brochure out there too that has some of this broken down. Yeah.
I have a question.
Yeah.
How does it work in the brain? Like, does it integrate into your DNA in the
Oh, thank you. Okay, no. So that's actually why I like this gene therapy. So AAV specifically, the gene stays kind of off to the side. We call it episomal. So it makes itself into its own little circle, and it kind of just goes off to the side. So unlike other types of gene therapy, so the AAV is kind of unique in this way. It doesn't go into your own DNA. It stays off on its own, which is part of why it's safer. And this is why, but this is also why we need to do this really long-term studies, because so I can prove that it looks safer long term. So what I can tell you is probably not. We have evidence. So there are some non-human primates where they were injected, I think it's over 10 years, and we were still seeing durable expression over 10 years. And then we've had one person there was a phase 1 study done back in 2013. So many years after he got treated, he had a back surgery and passed away from that. And we can see there, even then, so we're talking, I think it was like 4 or 5 years after, even then he still had durable expression at that point. So that's the best evidence I have right now. How do I show that in somebody who's alive? That's where it gets really hard, and that's where all the imaging comes into play.
If you're in the double-blinded trial, after you get the gene therapy, are you weaned off of the carbidopa levodopa, or are you staying on that for the length of the trial?
So my goal is to really not change it. So this is not meant to remove it. However, we did see that some people were able to significantly reduce their medication. This is person dependent, and this is part of why it's tricky to study Parkinson's because of this, because like the meds change, they'll automatically change everything I'm measuring. So the goal really is like try to no change as best we can, so we can see what's actually happening to GDNF and not the medicines on top of everything. That being said, patients really want to come off meds, as you can tell. So it's a bit of a battle right now. It's something we're actually trying to bring back to the FDA too.
When you put in the new genes,
Sorry,
exactly what do they control? What bodily functions or bodily parts?
I'm sorry, where are you? Oh, okay. So yeah, no, all we're adding in is a gene for GDNF. And so it's just
What,
one
what,
little
what are
snippet.
they for?
So GDNF it's called a growth factor or a neurotrophic factor. So trophic meaning food. So it's literally food for dopamine neurons. And what it really does is it is a way of, when GDNF is present, it says to the dopamine cells, everything's okay, you're all right. I know you're stressed over here, but we got you over here, it's okay. So it does seem to kind of quiet the immune system down. It seems to help like manage alpha-synuclein a little bit better. We actually see improvement in dopamine levels after GDNF on top of that. So, and I think it just allows the dopamine cells to just function more normally when they've got that little extra support with the GDNF. Does that? I'm not sure if that answered, but hopefully get an idea. Okay.
Yes.
Oh.
Yeah.
Are there comorbid conditions that would initially eliminate a client from being eligible for the trial?
Yes, participant. Like I feel very strongly about this. Is your people are handing over their lives, right? So this is volunteerism, and I really appreciate it. So, but yes. So again, if there's like straight up dementia, this is just not, it's brain surgery. This isn't the right study. If there's anything that's going to get in the way of us being able to interpret what we're seeing from the data, that's probably another good reason. If there's something in the brain that would make you not safe to have brain surgery, and we can only sometimes know that if we've got the MRI. So that's why there's so much in the screening. Is just we want to make sure it's safe more than anything. Yeah.
Where do the genes come from that you are putting in someone's brain, and is there a possibility that the body would reject those genes?
Oh, God, those are such good questions, Kara and Juwan. I hope we're getting all of these. So it basically is synthetic. So it's not coming from anybody. Like we've known this sequence of this gene forever. So we basically were able to just kind of put the A, G's, T's, and C's in the right order to make GDNF. So it's all synthetic. It's not coming from anyone.
Does this work better on men or women? What age group, left-handed or right-handed people?
I hope I have enough data someday to answer those questions. I just don't have the data today. I wish. There's a lot of really cool work with the Michael J. Fox Foundation that's looking at a lot of these other factors, and not necessarily with this treatment, but they're interested in those kinds of things too.
I have another question.
Yeah.
Do you think it's actually reducing inflammation in the brain, like you talked about the GDNF and inflammation? Is that maybe a mechanism?
I think so. I don't have a lot of data to back that up, but we do see, at least in primates, there's less microglia. I mean, it's just the overall inflammatory tone seems to be just a little bit calmer. And we do think it is one of the reasons GDNF works. GDNF is so kind of above everything else. I think it has such a broad effect this way. Inflammation just being one of them. Yeah.
Thank you.
Yeah.
How long has this study been going on?
So the very first study was started in 2013. It wrapped up in about 5 years, so 2018, 2019. There are some publications on that. And then the study I just showed you, that started in 2020 because that was the best time to start a clinical study. And it's still going. But we're about to wrap up. We'll have our first patient through their 5 years in August. So it's still going but wrapping up. And then the phase 2, we just opened middle of last year. So that's still very early on. So if anyone's interested, please, we want to talk to you. Yep.
Do you do any work with animal models?
So everything I showed you was built off of a lot of animal work, decades of animal work. I mean, lots. And so this, I don't know how we would have done it without it. I think there's one over here.
Do you know how much this costs? And is it covered by most insurance?
So we aren't approved yet to be able to even have that conversation. But you know what? This is important to us, and it's accessibility. And so it's an ongoing discussion that we are pioneering the way here for gene therapy, and gene therapy for something as big as Parkinson's. This hasn't been done before.
Any other questions?
There's one.
Oh, I'm
Okay, a few more.
coming.
We have
And then-
time for 2 more questions.
And I'll be here for a while, and we got a table out there too.
I do not understand the total dynamics in the studies, but for the people that participated and were placebo, do they have the opportunity in future studies, providing they're still in the criteria, can they participate? In a
Good
real,
question.
and as a real, they're getting the treatment as opposed to randomized and maybe being a placebo again.
So yeah, I feel very strongly about this. So if you were in the study and it looks like we're not having any major safety issue, we are making sure everybody is offered active treatment after we're able to unblind. That's important. At one point, the FDA didn't want us doing that, but I think that we were able to work with them and they understood that this was important. As long as we don't have any other new safety thing pop up. But if someone were in the placebo group, and it's a control surgery is what we're calling it, and they didn't want to continue on, then yes, they would still be eligible for other things too. Yeah. These are great questions, guys.
Yeah. Okay, we have time for one more question, if there is one. Any more questions? Okay, let's move on. Oh, one more. Okay. Oh. All right.
Can you yell?
This will be our.
A question on cost and
For the study?
for the study.
Okay, so I'm gonna-
How's it covered?
Okay, I'm going to get in my soapbox here for a second. Any legitimate clinical study should be on clinicaltrials.gov, and you shouldn't be paying a penny. Any legitimate clinical trial you shouldn't be paying to get into, okay?
You should be
So
paid.
we cover this. You are donating and volunteering your time and your brain. That's all
Okay.
we need, okay? So there's no cost.
And it's covered by Bayer?
Say again?
It's covered by Bayer?
Essentially, yes.
Okay.
We are the sponsor. We pay for this, and this is our
Okay.
duty.
Thank you.
Okay, thank you, Dr.
Thank you.
Van Laar. That was wonderful.